when I was a fellow back in 2007, we had the opportunity to present twice a month to this conference for the Neuropath CPC.
Don't even go there, Dirk. Okay?
Oh my Lord, that was trial by fire for sure. So I'm really excited. I was here a couple years ago and talked about CTE, and so I thought I'd take a little bit different tact for this talk. And at the end, I'll introduce you to a couple of really new activities that I think will lead to hopefully future Grand Rounds presentations around TBI and cancer, but we'll get there. So the majority of the talks will be focused on our efforts in neurodegenerative disease, and basically this is a summary of the neuropathology core. And so I'm going to go from there. So if you have any questions, let me know. This is a neurosurgery, very astute audience, and so I don't think I'll have to spend a lot of time on a lot of these. But as you can see, we have a very nice brain here. There's another brain there, right? That's a mouse brain. And so the point that I want to make is that human neuropathology, human disease, really establishes relevance, right? When we see things that are changing in human tissue, that tells us about biological relevance of a change. We can't really test for mechanism in human tissue, not from an autopsy perspective. We can do this in experimental systems, and I use the mouse as an example. The induced pluripotent stem cells and other stem cell technologies are a really promising new approach to test for mechanism in human tissues. But generally, we think about human tissues as establishing relevance, mouse tissues as helping us test hypotheses generated from those human tissues to test for mechanism, and those both basically inform each other. That's science in a nutshell. That's the focus of what we do in my lab, in the Neuropath core, in the neuropathology division, and I think broadly across much of what we do as physician scientists. And so we're going to spend all of our time today talking about the human tissue side and what we're trying to do to really maximize the amount of information we can get from the human brain when we see it at autopsy. So, key concepts. I think everybody here knows what neuropathology is. I think everybody here has seen brains. We take brains, we cut them up, and we look at them under the microscope, as you know. Neurodegenerative disease, really important conceptually, is it's progressive, right? Neurodegenerative diseases don't reverse, and they're classified usually by a specific lesion. And so I'm going to focus on this because you guys may not spend a lot of time thinking about neurodegenerative disease in your practice and that sort of thing. So we start with Ramoni Cajal, the father of neuropathology, who did, as you know, these just amazing, beautiful drawings of basically Golgi stains. These are silver stains, special stains that he developed way back in the day that were then used by Dr. Alzheimer, Lois Alzheimer, studying the first Alzheimer's patient, Auguste D. She presented when she was 51. So this was an early onset Alzheimer's disease case. We're not really sure if it was really sporadic Alzheimer's disease or some familial variant. But Dr. Alzheimer described these amyloid plaques and neurofibrillary tangles using the staining technology basically developed by Dr. Cajal. There was a major advancement some decades ago called immunohistochemistry, where we could take antibodies and specifically label things like tau. You can see the neuritic component of a neuritic plaque in these neurofibrillary tangles using a phospho-tau antibody, and then of course this is a beta amyloid stain showing a dense core plaque and some cerebral amyloid angiopathy, which we actually see from you guys once in a while. That is the state of neuropathology today, right? That is the state of neuropathology today, and I'm going to emphasize that. There's two critical concepts about neurodegenerative disease. They're characterized by pathologic peptides almost always across the board. Alzheimer's disease, of course, has phospho-tau. It's not exclusive to Alzheimer's disease. And then beta amyloid plaques define Alzheimer's disease pathology. Lewy bodies and Lewy neurites are the characteristic features, the diagnostic, the pathognomonic features of Lewy body disease and Parkinson's disease. And then, of course, we have these TDP43 inclusions, and old school would be Benina bodies and other things that were characteristic of ALS, and some of the FTLDs, certainly the FTLD TDP would be characteristic for TDP lesions. The other critical concept, and these are important because they will tell us, they are telling us about how these diseases work, how they develop, what's the pathophysiology of these diseases. So in addition to having these typical pathologic peptides associated with the diseases, they're characterized by selective vulnerability. And so what does that mean? That means that parts of the brain are affected before other parts of the brain. And if we can understand why that is, why these peptide deposits start in one place versus the other, what makes that region vulnerable, I think that's going to give us insights that will end up helping us solve these things and develop therapies and preventive strategies. So tau starts in the medial temporal lobe and works its way up into the neocortex. Beta amyloid starts in the neocortex and works its way down through the deep cerebral structures, brainstem, and then ultimately ending in the cerebellum. Lewy body disease starts usually in the brainstem and limbic structures and then works its way back out into the cortex. And TDP-43 is kind of the odd duck in that we have many different sort of TDP-43 characteristic lesions, but your typical FTLD TDP starts in the brainstem and temporal lobe and works its way out into the cortex. That's the ALS type of TDP. And so understanding selective vulnerability, I think, is critical to understanding these diseases. And one of the goals that we have in the Neuropath lab is to try to compare what's happening in regions that are vulnerable versus regions that are less susceptible or resistant to developing pathology. The other really critical concept, I think, that is a major focus for us across the board. This is not just Alzheimer's disease. This is not just neurodegenerative disease. This works for cancer. This works for everything. In that there are different states of neuropathology, and as a neuropathologist, I try to incorporate clinical function at some level, but there's an important difference between whether a tissue is diseased and whether that affects the phenotype of that person. And so if you're healthy and you don't have any pathology, then that's great. Hopefully that's all of us, right? I guarantee, after looking at a whole bunch of young people's brains, that a lot of us in here have some level of tau in our brains. And so I don't know that we would necessarily classify as healthy or resistant versus whether we have some sort of a latent or preclinical disease. Hopefully none of us have high pathology, right? Certainly if we're impaired, if we're demented, in the case of Alzheimer's disease, and we have high Alzheimer's disease pathology, that's penetrant injury. Simple, right? It's this area that I think is tremendously fascinating. If we get very old and we have low pathology, then that would be, we somehow have evaded the processes that cause us to develop these pathologies. It's classic for Alzheimer's disease. People who are over 85, lots of people have Alzheimer's disease. If people over 85 or over 90 don't have any Alzheimer's disease change, there's something special about those people. We think of them as resistant. The other side of the coin is the people who have a lot of pathology in their brain, which normally in most people would cause some impairment, but they're not impaired. And it may be cognitive reserve is another word for this, but we are really interested in comparing people who have lots of pathology and are demented versus people who have lots of pathology and are not. And that could go for the same as for traumatic brain injury, right? You can have someone who has a severe traumatic brain injury and they're reasonably high functioning versus another person who has the same or even less of an injury and is debilitated. What is the difference, right? I think that's really critical to understanding these diseases. And so these are the sort of concepts that we are focused on in the Neuropath core. And the gap in knowledge that is sort of the focus of everything we do is to try to understand the pathophysiology of these diseases and mechanisms of resistance and resilience. So the Neuropath core, I'll just give you a quick history. Some of you will remember these folks. So the Neuropath core really began in 1985 when the Alzheimer's Center was started. I have to give huge props to Eric Larson and Bud Kukel, who were really around at the beginning of the time for this, as far as this goes, and envisioned sort of developing, along with George Martin, who's still around and is at 90-something, is still getting RL1s, who built the concept around the Alzheimer's Center and the ADPR, which became the Adult Changes in Thought Studies, which have been around since the 80s. The Neuropath core was started then, and it was really the visionary leadership of Jim Leverins and Dave Notchlin who many of you will probably remember, some of you, maybe not that many actually, as the neuropathologist. Jim's actually a neurologist with neuropath training, who really started neurodegenerative disease neuropathology research here at UW. Of course, that was heavily supported by Buster, and to this day, Buster is supporting this work, which is his lasting legacy in one of so many ways. Just yesterday, I was down in the autopsy suite here in this building, cutting off pieces of cortex and cerebellum from a couple of the early presenilin 1 mutant brains from those early cohorts. So we're still using those brains to this day, which is really truly amazing. And then in 2002, Buster and Rich and others recruited this guy to the UW, and he brought with him a cohort of fantastic people: Randy Wolcher, Jay-Z, who everybody knows, and then Amy Shantz, who all came from Vanderbilt. Tom recognizes the power of neuropath and epi, right? That is a concept that really escaped people for a very long time and is now really driving a lot of Alzheimer's disease and related dementia research, and built out of the ACT study. He saw the value in a population-based cohort to collect neuropathology in a way that enabled us to not just do stage, but to actually leverage the biochemical expertise that he and his team brought to really start to develop quantitative approaches to studying neuropathology. And so he instituted rapid autopsies in which we try to get the brains out fast so that we can get frozen tissue, and that is useful for things like biochemical testing for isoprostanes and other things you can't do with fixed tissue. He also instituted standard sampling and standard staining protocols so that we have uniform assessments across the neuropath core from the earliest brain. That really set the stage for the NP core to have a national impact that was led by Tom, and so we're all grateful. Tom recruited these two guys, Josh Sonnen and myself, to participate, to help these folks in the neuropath core, which was wonderful. And then one of the last things that Tom did was really help recruit Caitlin Latimer, who you all know. I think anybody who does Frozens knows Caitlin, and knows that she's just amazing and is in line to take over this thing someday. So, what is the mission? So, the neuropath core mission, as it was developed, is to support brain aging, injury, and degeneration research through multiple sort of aims. Of course, of primary importance is to build a repository, right? And we, I think, are doing a really good job of that. We continue to develop those methods and those protocols to ensure that we are one of the top repositories in the country. We're certainly one of the busiest, and I'll show some data about that later. Of course, we need to provide diagnostic expertise. When a person comes from a study and donates their brain, the brain is not useful if we aren't able to tell the study about that brain. And that's what we do, and we do that at or above national standards. We maintain a rich training environment. I can't emphasize how important this is. We try to count how many trainees we have coming through every year, and every 5-year period for the Alzheimer's Center, it's around 200 people that come through and are exposed through week-long rotations or longer rotations, or some iteration of our training program through our neuropath core. Many of those people are leading their own neuropath course. I am a product of this training environment. Caitlin Latimer is a product of this training environment. Josh Sonnen, Randy Wolcher, Jay-Z are all products of this training environment. It's a real statement on making sure that the legacy is continued and that science is really supported. One of the things I'm most proud of, and that's something we're working with Christine McDonald here, who's obviously famous, to really push the envelope on how much information we're collecting from these brains, right? I appreciate philanthropy, I appreciate collaboration, I appreciate all of the different great things that people do for each other in science, but I truly believe that the greatest gift you can give to science is your brain. It is who you are, right? It is what makes you who you are. And I am humbled every time we're down there taking a person's brain out and preserving it for future research. And so, to honor that donation, one of the things that we try to do is continue to innovate, to continue to push the envelope on the utility of that tissue for generating scientific data to help understand these diseases. And so that's a really important component. Of course, we want to foster mechanistic research. We do not generate mouse models, but hopefully we generate data that will help inform mouse models. And lately, we're doing more and more cell and tissue preservation that promotes stem cell models and cell lines that can be studied directly from these patients. And then finally, we promote local and national research. Of course, we share a ton of tissue and data. It's thousands of specimens a year with all of the associated data. But Tom and now myself are involved in many different consensus conferences that are helping derive the national protocols and guidelines for how we do these, how we characterize Alzheimer's disease, CTE, that sort of thing. And so we're real proud of that. So I'll just go through our protocol. Normally I warn people about this, but you're all neurosurgeons, so I'm not too worried about that. The way we used to do this is we would take the brain fresh. If we had a postmortem interval less than 8 hours, we would take one hemisphere, we would dissect it basically by hand. We would take these samples from M and above, is traditionally was what was started in 2002. All of this sort of developed over time, mostly in the last 6 to 8 years. But you can see that we're taking a lot of samples. There's 70 or 80 different samples of frozen tissue from every brain that were taken from the thousands of brains, or the 1,000 or so brains we have rapid autopsies on. And then of course the brains are fixed and cut, and then that was sort of our standard protocol. I can't emphasize how important this is, and I think I have another slide in just a minute, but this protocol is what was used because Tom was the co-chair of the NIAA conference to basically define the neuropathology of Alzheimer's disease. This protocol, developed at UW, was what was used to define the criteria. So the current criteria since 2012 for everybody in the world to basically diagnose Alzheimer's disease, neuropathologic change, and related dementias are derived from UW. And it's fantastic. It's a fantastic legacy. And so we didn't have to change hardly anything when those protocols came out because we were already doing it. That may be Tom's greatest legacy, I think, is really developing national standards based on the work that was done here. So again, this is just a list of all of the samples we take at rapid autopsy. For any of you that might be interested in brain research using flash frozen tissue from many different kinds of cases, mostly old people with or without dementia, these are available for your research. And we share this with people weekly. We have shipments that go out to people. The diagnostic expertise, of course, we do what we're supposed to do by the national guidelines, which, because of the limitations of what the neuropathology community can do, are still based on histochemical stains, right? These are the same technology that Dr. Alzheimer is using, and immunohistochemistry and standard H&E stains. That is basically the state of the art for standard diagnostic protocols. These are the criteria I just mentioned that Tom and Brad Hyman from MGH co-chaired and developed that we're still using today. That is the current state of the art. That is the current accepted guidelines that we use for our diagnostics. By the way, if we end up with a surgical specimen from you guys, from somebody who's old, who had a spontaneous bleed or something like that, maybe had a tumor, often we will do some assessment for Alzheimer's disease neuropathologic change. We cannot complete this assessment. You need to have multiple brain regions, some of which you're not going to take in surgery, but we will try to apply this in some way to your surgical specimens. And so if you ever see that, let us know, and we're happy to sort of work through that. And we not only diagnose all the brains that we have, but we have a diagnostic collaboration led by Dr. Marshall, Desiree Marshall, who I'm sure you all know, that provides consult services for the community, and she and I both serve as liaison for people who may not be in our research studies, but who may have a relative who is demented, and they want to know for their family and for other reasons what happened. And so we don't just perform, study the brains that are donated to us. So how do we do this? We have a reasonably unique way of cutting our brains. Most of you have been on brain cutting. You go through the UW with us when you're on our rotation. We've all trained, most of the people who are from here have trained with Buster. We don't cut stuff by hand. We use this brain slicer, which you might recognize for other reasons. And we basically embed our brains in agarose. This is just the same thing we've done a DNA gel on. It's cut on our brain slicer, laid out, and photographed. And again, this is the standard protocol that we still do to this day to maintain continuity with all of those cases from our historical perspective. Whenever we develop some new advancement, we don't alter what we have been doing for a long time so that there's continuity of data across our entire cohort. That is really critical. When we add stuff, we have to figure out how to go back and do it on everybody else. It's important, and that's a constant issue. We generate a neuropath report, and these reports are either sent to researchers in the cohorts or to the family directly in some cases. And this is just the latest diagnostic line. The report's usually 4 or 5 pages, but we try to hit all the high points for people so they know the things that can cause dementia, whether they're present or absent. And then, of course, we have a common area here for things. You know, we get incidental meningiomas routinely and some other things, meds once in a while that go in there. So next I want to summarize a little bit about what impact the NeuroPath core has had. Early on I talked about Jim Leverins and Dave Notchlin and Eric. You know, this is one of the first studies that came out in a community-based sample. Not a highly selected Alzheimer's disease cohort, but a community-based sample focused on neuropath. Really, this was really groundbreaking at the time. There have been multiple other important papers coming out. ApoE genotyping. This center has always been very famous for its genetics. ApoE genotyping in the community-based sample was really a critical development. Jim Leverins spent a lot of time studying hippocampal sclerosis, and I can't tell you how important this is with respect to the work that we do. Right? People who consent to autopsy are not the same as everybody else. Right? There's something about a person who consents to autopsy that there's something about them that makes them want to do that. And there's things that make other people not want to do it. And to study that, to understand those differences, you really need to be in a representative population. And some of the early work that came out from Jim and Debbie and others really started to help us understand the information we're gleaning from these autopsies needs to be qualified by the variables that go into having someone donate their brain. Josh Sonnen, you all probably know Josh. He's now at McGill. Josh did his fellowship here. He and I started the same exact time. And Josh published some really important papers focused on the pathological correlates in the community-based sample. So really, by the time the ACT study was at 200 or 300 people, Josh and Tom were doing this neuroepi work in the ACT study. And this Annals paper was Josh's fellowship, the paper that came out of his fellowship. It's cited a million times, and it's really been impactful. He also has studied changes in people with diabetes, and then he published this paper. I can't ever imagine having a better title for a paper, which is basically just characterizing what is the typical pathology in community-based samples. It's a really nice study, and he focused not just on people who are demented, but on people who are not demented, and started to get toward that sort of resilience category. What he did was really interesting. He made this. It's basically a summary score where blue is Alzheimer's disease pathology, orange is vascular brain injury, and specifically microvascular brain injury, and green is Lewy body disease. And he just gave each 3 points based on the severity and added them up, and found in the first 116 ACT participants that there's a high prevalence of mixed pathology. This is of great interest today. People understand that, unlike in an Alzheimer's center where you have people with reasonably pure Alzheimer's disease, in the community, all of us, right, as we age, we're probably gonna get multiple things to happen to us. And we need to be studying those kinds of people to really impact society. And this is used by, this type of a system is used by many people to this day as a way to judge. It's not based on any sort of quantitative metrics other than the standard scoring, but it's really great. And all this is basically saying is that basically the more pathologies you have, the more summary neuropathology you have, it predicts that you're more likely to be demented, right? And that makes sense, but it needed to be shown, and that's what he showed with this data, which was great. So the neuroepi, I'm just going to run through a bunch of, just to highlight all of the work that's been done. There are hundreds and hundreds of papers. They've studied atrial fibrillation, blood pressure, and autopsy cigarette smoking and Lewy body accumulation, which was verified in this, that there's a reduced risk for Lewy bodies with smoking. One of the few things with cigarette smoking seems to help. NSAIDs is a big issue. There was actually a clinical trial based here from some of this data that failed due to the toxicity of the drugs without really getting to the point where we could see if it affected dementia. Statins is a very important study that was published by Tom and his groups. This is something that we're all very proud of. This was something really initiated by Rich and Tom way back in the day, and it led to our Allen study that I'll talk about in a minute. But Paul and Eric went into the ACT study and the Rush studies and looked at traumatic brain injury with late life neurodegeneration and found not an association with Alzheimer's disease, which is what everybody thought, but an association with Parkinson's disease and Lewy body and incident Parkinson's disease. That was then verified in 90,000 people in Kaiser, California. It's a really groundbreaking study that I was really proud to be a part of. Paul has published several papers on glucose, including a New England Journal article. This is the one that focused on neuropath and glucose. And of course, all of these things, blood glucose and diabetes, hypercholesterolemia, hypertension, all of these things contribute to this mixed pathology that we generate as we age. Really important work coming out of this group. And so lots and lots of studies, boy. And then so more recently, this is more recent work that I've been involved in that has come out of our group. There's a major focus on genetics now. Our Neuropath core has contributed hundreds and hundreds of cases to the studies that are using GWAS and other things. Tim Homan is looking at sex differences in all kinds of different parameters, which has been very impactful. This is work that is basically done with Jeff, really driving this. I don't know where Jeff went. Jeff Ogeman and Andrew Coe, Dr. Ellenbogen, and others with some of the surgical material that uses the infrastructure of the Neuropath core to make sure that the tissue coming from your ORs is making it to the Allen Institute in a very rapid and safe way. This is a seminal paper that was just published in Nature that I think we're going to hear a lot about, comparing the cell types in mice and humans, and showing that generally they're very similar from a broad perspective, but when you start looking at the different, very defined areas, like what layer of the cortex are they in, what is their morphology, who do they connect to, there's really significant differences between mice and humans, which may underlie some of the problems we've had in translating clinical trials for Alzheimer's disease from mice into humans, which has been a giant failure. This was a recent paper from collaboration with folks at Minnesota, which I think was the first time I ever published anything with my Minnesota friends since I left, which was great. This is a paper that just came out with Stanley Prusner's lab, focused on A beta and tau, and how the propagatory properties of these pathologic peptides, like the prion-like properties, change over time. It actually gets less propagable with age, which is really fascinating work. Carlo is a genius. And then, of course, Dr. Prusner is really amazing. And then this is a recent report from a consensus conference describing limbic predominant age-related TDP-43 disease, which you're going to hear a lot about. This is not a new disease, but it's a new name called LATE. It's highly prevalent. It's the focus of Caitlin Latimer's work here, and I think it's going to be the number 4, right? We have AD, vascular brain injury, Lewy bodies. Late is going to be number 4 when it comes to contributions to dementia in the elderly. And then just a JCI article with Tal Ma down at Wake Forest, which we collaborate with. So our productivity, this is a little dated now, but we have 3,500-ish brains in our repository. Those are all stored in this building. Some of those are from the Alzheimer's Center. We are up to about 880 from ACT now. The Seattle longitudinal study is really important. We have obviously a lot of different tissue. I present this to groups in the hope that people will think of us when they're doing research, and they think about maybe adding some human tissue component to their research, maybe as a validation for something they're looking at in their mice. We're making cell lines. We have a lot of historical or legacy fibroblasts, and of course DNA from a lot of people. This is our productivity the last 5 years, which I'm very proud of. I took over in 2013, and generally we've been doing quite well as we've progressed from both a publications perspective. This is the number of requests we're getting for tissue. This is the number of tissue samples we share each year. Right? That I'm more proud of than anything. Right? I mean, last year alone we gave away over almost 8,000 samples of tissue to researchers around the world. That's really, I think, important. We'll see how this is impacted in 2019 with the closure of the R&D building. Guarantee this is going to go that way, but hopefully that'll be mitigated sooner than later. This is the composition of our cases with respect to old people. We have, this is a really nice sort of derivation of what Josh had developed with the summary score that Caitlin Latimer put together. We have our non-demented people in our bank, and we have our demented people in our bank. And then this is the group of MCI, it's just small. This is the summary NeuroPath score, just like Josh showed, and a couple things are important. You can see that Alzheimer's disease affects almost everybody, right? Almost everybody in this bank, and these are old people, has some level of Alzheimer's disease, right? So it is highly, highly prevalent. So if you get to be really old and you don't have any Alzheimer's disease, there's something really special about you. I'll tell you about that in a second. But we just randomly put this line at 3 to sort of show the concept, with the idea that if you have pathology below this level, that's really low pathology for that age, and that is you're non-demented and you have low pathology, then that's expected, right? You shouldn't be demented. But if you have a lot of pathology like this, like these people, and you're not demented, there's something really special about it. The same goes over here. If you have, although it's kind of the opposite, right? If you're demented and you don't have much pathology, then we're missing something, right? I don't know what we're missing, but we're missing something. Or you're just extraordinarily vulnerable to a little bit of pathology. Not as much of an area of interest to us, but certainly something to study. And then, of course, you get a lot of Lewy body disease in here and vascular brain injury. And then you can just see the sort of the range of our demented and non-demented people. There's a ton of overlap. There's just huge opportunities to study these people if you're doing any kind of research. And so that derived a study by Caitlin Latimer, which I think is spectacular. She looked for resistant people, which she defined as greater than 85, Brock stage less than 4, and no plaques, and resilient people, which were people that were Brock 6, which is the highest Brock stage, and frequent plaques, which is the highest plaque stage. And then her controls were demented, right? These are non-demented. Her controls were demented, also high path, right? And so when we looked through the whole ACT study, we found 7 people who we categorized as resilient. So these are 7 people that were not demented and had the highest level of pathology that we could define by current standards. And we found 14 people that were resistant. We matched those people by age, sex, pathology in the case of the resilient folks, and we didn't pick the pathology to match. And what you see is what is, I think, going to be really important for you to recognize going forward in your old people, in your old patients, is this high prevalence of TDP-43 pathology in demented people. This is late. This is this concept of limbic-associated TDP-43 disease. We don't see much of it in resilience or in resistance. We just don't. In fact, these 2 people were really, really interesting to find that they were there. We had selected based on Alzheimer's disease, but we didn't select on the other things. And so what you find is in resilient people, they have less other comorbidities, but TDP-43 tops the list. Same goes for resistant people, right? They have less pathology, but we selected on pathology. I think that's a fantastic demonstration of out of 800 some people, we only found that many that were resistant and resilient by stringently defined mechanisms. And then Caitlin went and quantified tau and TDP-43 and showed basically a strong correlation. We obviously are going to see a difference between TDP-43 because most of the people didn't have it who were resilient. But the resilient people also had less tau. That highlights a huge problem that we have in neuropathology, which is the current criteria are ordinal grading criteria, Brock stage, CIRAD score, fall phase, right? 1 to 3, 1 to 5, right? It is woefully inadequate to really describe the burden of pathology that happens in people. And we need more quantitative methods to really get to that. So Caitlin did that. That paper was just recently published this year and has had a really great impact in our societies. So I'm going to transition now to tell you a little bit about what we are doing next. So a few years ago, when Tom left, I took stock of the whole situation and decided it was a great opportunity for us to sort of rethink how we wanted to do this without compromising the protocols that we had been doing for so long and continuing to maintain that. But what can we do to really push the limits? And so some of the things I just built out of what Tom had done, and some of the things we're doing are relatively new, and I'll tell you about that. But generally the way that I'm thinking about this every day is our standard practice is we have well-characterized cohorts that we can basically do traditional neuropathology, right? That's our standard practice. That's what most of the Alzheimer's centers are doing. State of the art are really more quantitative approaches, and I'll get to a study that we did with Dr. Ellenbogen looking at transcriptomics, the Allen Institute, but you know, these sort of grind and bind type of approaches to tissue. That's why we've been freezing tissue for so long. That gives you a much more quantitative way to look at pathology. But where we really want to go, and what I think will be really transformative, are single cell applications. Some of the molecular profiling that Dr. Patel, Dr. Stamino, and others are doing in cancer, the Allen Institute's doing. That's where we want to go with neuropathology, right? We need away from plaques and tangles. We need to start understanding cells that are vulnerable before they have a tangle, before the plaques are deposited. What is happening early in disease, and how can we characterize that pathology to better demonstrate the disease as a diagnostic, but also to understand what's happening early so that we can develop hypotheses about mechanism and interventions. So that's where I think we need to go. We've developed, we're trying to deploy certain quantitative approaches. We can do slide scanning and image analysis. We can do unbiased stereology. These are not new approaches. Tom, along with Nadia Postupna and I, developed Histolide, which was basically an ELISA on a paraffin section, which I think was really transformative and has been a very nice approach that we've used in our lab to generate quantitative data on pathologic peptides. And then with frozen tissue, we have your typical biochemical assays, immunoassays. We have molecular assays, and then we started focusing on synaptosomes and taking regions of brain that were relevant for functional imaging in dementia quite some time ago. Those are the sort of quantitative approaches that exist that we've applied. So I'll tell you a little bit about each of these. Histolide, as I said, is basically an ELISA. So instead of staining the tissue right away, you basically bind the antibodies and then put in a soluble chromogen reaction with alkaline phosphatase that turns the solution a different color, and using Beer's law, you can quantify how much binding there is based on the colorimetric change. And then at the end of it, you stain it with an insoluble chromogen, and you get a nice histologic stain. So that was a really, really nice, simple, but very nice development that Tom used to study the ACT study. And Nadia really drove this, and we found that we added about 10% predictive power to whether a person is demented using this quantitative approach to measure tau and A beta versus the typical Brock and CIRAD score. It was quite quite helpful, and we've continued that to this day. There have been several other studies published with that data set, which we stopped collecting at around 500 cases because we transitioned to a new one. Our modern BioRepository, so we revisited how we're collecting brains and came up with a new way adapted from the Allen Institute. And so what we do now is we take half a brain, we strip the meninges off of it, we then put it in this custom-built sort of mold where you can see the brain and the cerebellum is put on the edge, and then we put this blue stuff. This is dental cement. It's basically alginate. It's great. It smells like peppermint, right? The whole room smells like peppermint. And we embed the brain into this block. It fixes in about 5 minutes, probably less. Actually, if you're not careful, it fixes too fast. And then we can cut the brain using this custom jig into 4 millimeter thick sections. Cutting the fresh brain into 4 millimeters, it's hard to cut a fixed brain in 4 millimeter thick sections, and this is done very, very nicely to where you get this beautiful, uniformly thick, very nice slice of brain that we lay out, we photograph, and then we freeze it based using, this is an isopentane slurry, a sort of a super cooled isopentane slurry. And what that does, using this Teflon-coated plate, is it freezes the tissue very uniformly. It prevents it from getting the ice crystal damage that you get from flash freezing in liquid nitrogen. We sandwich it, flip it over, and get both sides fixed equally nicely, and then you get a beautifully frozen piece of brain that we can then go back to, in which the cells are very, very nicely preserved. And preliminary studies from these brains with comparable flash frozen tissue from the adjacent area shows that our RNA quality is much, much higher. We have RIN values in the 9s from these, which we did not expect. We did not expect that, and so if that holds up, this is going to be hugely beneficial. So this is how we're doing our brains now. So in addition to having really high quality frozen tissue, we have the whole brain to work from. Before we were just taking samples. We have the whole brain now. And this is Christine's work, which is how many brains now?
175.
175 brains have been scanned. So I showed you this is the old school. Christine and Lisa in the lab have worked really hard to develop better ways to embed these brains in agros to avoid air bubbles. In fact, I think on Fridays it's like bubble popping day or something. Anyway, the brain gets embedded in this block, which you can see here. They color code by using dyes so that they know which brain's in the magnet at what time and they don't mix them up. And then there's the team put with the brain going into the MRI and then the results, right? And all happy all the time. So every brain is getting imaged, which I think is spectacular. This is a 25-year-old bull rider that died. You can see that from the imaging. You can see these lesions, I believe. Let me see. So was this the antemortem here? Or was that postmortem? This is postmortem. So you can see these small lesions. That Christine is picking up, which we would never see grossly, right? We would never sample this pathologically, and we're getting it. This is the antemortem for the bull rider, where he had a pretty significant injury. This is the postmortem MRI, where you can barely see it, and pathologically we would never sample that. But we did, and sure enough, there's old traumatic axonal injury that we were able to detect thanks to the imaging. We're applying this to every brain. Christine is now down at brain slicing and sampling every day, every time we do it with her laptop, and we go through the brain, for every brain, sampling for gross pathology and sampling for imaging. We're finding all kinds of new stuff. This is where we should all be from a neuropath perspective. If we really want to define the injury to the brain, we should be doing this. I'm gonna go reasonably quickly through the next few things. This is how we embed our brains. I just showed you that. That's our brain slicer. They're all laid out. We have them all photographed, and we're using this Amira software to basically reconstruct brains that didn't get an MRI. So this is an ex vivo MRI from Christine's lab. This is a reconstruction based on the photographs that have been reconstituted into basically a virtual MRI. The goal is for these to go into FreeSurfer to get the volumetric data, just like Christine. We have over 1,000 brains archived with these photographs, evenly sliced, that we could generate volumetric data using this technique, and then they go and they encode the different regions. And you can see, comparing ex vivo MRI, FreeSurfer versus what we're doing with our manual constructions, a very strong correlation with the volumes we're getting. And so we're really excited that this is going to be another way to study these brains. This is our new sampling protocol. This is what Tom did. This is what we've added, right? We've realized that there's a ton of things we're not sampling. This is all based on neuroimaging. This is all based on traumatic brain injury. This is all based on CTE pathology. And so we're really expanding the sampling so that we have a much better view of the heterogeneity of disease. And you can see here just a list of things that inform that sampling protocol. We're really excited about this, even though it's a lot of work. We're not looking at every slide on every case, but we're certainly sampling all these so that we can. And then this is another technique we're really excited about. You can take basically pieces of fresh or fixed brain, and you can put them through a protocol that uses cinnamon oil to clear it. This is like Clarity, but it's a cinnamon oil base, so the lab smells like cinnamon oil. We're not trying to get the lab smelling really great with all these, but so you clear these pieces of brain, you can see through them, and then you can stain them. This is a lectin stain, and this is actually a movie, and it takes 5 minutes to go from through the 4 millimeters of tissue. Looking at these, you can see all the branch points. We're really excited about the 3D volumetric studies we can do, and then this is a tau immunostain showing the neuritic plaques and the neurofibrillary tangles with the same thing. So we're hoping to understand more 3D relationships between vessels, between amyloid, beta amyloid, between tau and inflammation, lymphatic system through this technology, which we can't get in a thin section. And so that's what we're applying there. This is the next generation histolide that we've developed using Luminex platform. Basically, we're grinding up the tissue instead of doing it on the slide. And we have the opportunity to do a multiplex approach using these solution phase reactants or antigens. And it has been really, really useful in expanding histolide from a single epitope per slide to multiple. And this can be multiplexed to dozens of epitopes in one. This was just published. Sorry, I'm running out of time, so I'm trying to get to the end. We're taking the leptomeninges from every case with Jessica Young, who generates primary cell lines from these leptomeninges, which she can generate iPS cells and then neurons, and then she's also directly converting these cells into neurons. And so from every one of our autopsies, we have cells that can be used to study the genetic influences of that person. And that was just recently published. And so finally, this is how I sort of envision how neuropathology should be, right? We want to have really well characterized cohorts, neuroimaging and other biomarkers, genetics, clinical characterization, and then we want to deploy a really diverse and quantitative neuropathology battery of tests to generate as much relevant data as we can from that tissue, and then use that information to basically inform hypothesis testing and model building. That's the goal of the NeuroPath Core. This is a study that Rich really developed, right? I mean, it was you with Mr. Allen who developed the idea and funding for this study. This was a collaboration between us and the Allen Brain Institute called the Brain Aging, Dementia, and TBI study, where we basically did what we had tried to do. We took a well-characterized cohort, we deployed multiple different neuropathological approaches, and they did transcriptomics on 4 different brain regions to understand the long-term effects at the structural and molecular level of traumatic brain injury. And we didn't find much for TBI, but there was some really interesting data that is all publicly available and was recently published, not that recently anymore, in eLife. So, these are the different studies that the NeuroPath Core supports. I have 2 more slides and I'll stop. We have multiple other studies, but I want to highlight the ADRC as this could not have happened without the Alzheimer's Center. Right? The Alzheimer's Center really developed the infrastructure for us to get started, for us to develop our processes, and then the Adult Changes in the ACT study has really set the stage for what you can do with a well-characterized population-based cohort in neuropathology. I'll talk briefly about the Northwest Brain Donor Network and the Allen and the Neuro-Oncology Brain Repository, but there's a lot of different studies that contribute to the NeuroPath Core and that the NeuroPath Core contributes to. So this is a U19 that we've done with the Allen Institute. I'm leading, along with Tom and Eric, Project 1, and Ed is basically overseeing the whole thing, including Project 2 and Project 3, which is a center that's basically focused on single-cell profiling with single-cell transcriptomics and spatial methods, Alzheimer's disease. This got a fundable score. It's basically funded, except for the continuing resolution, they're not signing off on it yet. So we're really excited about that. That's going to generate, I think, some really important cell types data. And I would love to come back in a year or two and tell you how we're doing with this. The goals, I'm going to skip this just for time, but it's really focused on understanding vulnerable cell types early in disease in Alzheimer's disease. The Pacific Northwest Brain Donor Network is something that we started. This was a fellowship project for Dr. Marshall, Desiree Marshall, to develop a research platform that we wanted to use to study populations, people who are unlikely to be enrolled in studies, right? Traumatic brain injury really stimulated this. It really did. And so the model was to use site life and our medical examiners to refer patients to us who had died, and then to deploy all of the different applications that we have to understand these people. We have two projects. The first one was focused on military-related trauma. The second one is normal adult brain structure. So we're supplying the brains for the Allen Institute for their atlasing through this brain donor network. And to date, we have 58 cases, which is remarkable considering this is funded on a shoestring. The average age is 46, so we're generating young people. Christine is basically imaging every single case. We have 61% of these folks are military veterans, 76% are high school contact sports, and there's a bunch of these people with really well documented significant traumatic brain injuries. A lot of suicides. This is going to be a critical resource for TBI research in our institution. And then finally, with PJ Semino, we are using the NeuroPath core to start just this year the Neuro-Oncology Brain Repository. The focus of this, in concert with work that PJ and Anoop are doing, thanks to Rich and Manny and other people's support, to develop a neurosurgical tissue repository for the neurosurgical cases, to really understand gliomogenesis and glioma pathophysiology from the time it's diagnosed to death. For the Neuro-Oncology Brain Repository, the idea, and this is really PJ and Anoop and not me, is to focus on molecular and single-cell profiling of these tumors from the surgical resection, the initial resection, all the way through to the end at the autopsy for people who donate their brain. We want to understand the neuropathology and neuroimaging correlates in gliomas. We see cases where it looks like it's a recurrence and it's just reactive. It would be great to know better. So when we're not able to get a rapid on one of these brains, Christine has been very generous with her time in scanning these cases with MRI so that we can get those correlates. There's a lot of different mechanisms that we can study with this, and then I'm very interested in how gliomas and glioblastomas kill people and what kind of toxicity the adjacent brain undergoes from the tumor and from the treatments, and how we can better affect those cases, how we can better treat people based on this. We've had 9 cases so far. I'm not sure why. There's 8 of them are men, and they're all glioblastomas, including one person who just declined treatment, which I think is going to be extremely important. So with that, this is incomplete, but it's tons of people, and I've already told you how important I think brain donation is. So I'm going to stop. Thank you. Sorry, I'm 1 minute short of 8, so I should have left the time for questions, but thank you very much.
Christopher Dirk, superb. Any questions? You must have lots of questions. I was a little confused when you looked at both ends of the epidemiology, and you said the people that had lots of pathology, neuropathology, but didn't have the disease, you weren't interested at either end of the spectrum, or you are going to study, because those seem to be the most fascinating. That's the conundrum, right? Why there are people with incredible beta amyloid and they don't have the clinical disease.
Those are the people we were very interested in studying. Okay. It's the people who have dementia, but we don't see any reason for it.
Yeah.
That we haven't focused on. I shouldn't say they're uninteresting, and I definitely misspoke there. Those people are very interesting, but often it's something that we have a hard time defining pathologically, right, like alcohol or things like that. Exactly. Yeah.
Got it.
And we
Got it.
suck at that. But the people who have pathology and are phenotypically normal, hell yeah.
Yeah,
Right?
yeah.
There's something special about them.
Right, exactly. What are the genes that protect them?
Right.
So how
What did they
we-
eat? What did they drink? Where did they live?
Probably drinking heavily and smoking.
Right.
That probably rules. So, questions, any other questions? That was fabulous. That was unbelievable because, we're just right down the hall and, we think that everybody thinks that they're doing all this great work and we find out
Oh.
literally 10 feet away, there's spectacular work.
Everybody's
being
doing great work. So I would love to come back. I think there's gonna be a great opportunity to describe the brain tumor banking
Yep.
and the research that's coming from that. I think that, Anoop has really been, and PJ have been really essential in that. And then I would love to come back and talk about TBI again someday, because there's a whole story with the brain donor network that we need to tell, with respect to military TBI, with respect to, mild to severe long-term effects of TBI. But that's the neuropathy.